Is it good?
Great. Okay. Okay. So it's a pleasure today to introduce Maxime Galloy, who is a PhD student in Amélie Frade-Turcot's laboratory in the Department of Molecular Biology, Medical Biochemistry, and Pathology at the Université de Laval in Quebec, Canada. And the laboratory is studying the DNA damage signaling and repair pathways in different conditions, including replication stress. And Maxime specifically is interested in deciphering how chromatin composition surrounding DNA damage impacts the repair. So now I will turn it over to you, Maxime, to share your work with us.
Thank you. Thank you for the introduction. So, hi, everyone. Thanks to the organizer for giving me this opportunity to present my work today. So I'm a PhD student in Amélie Frade-Turcot's laboratory, and as introduced, I'm studying how chromatin and the smallest subunit of the chromatin here, the nucleosome, could impact the DNA damage response. And so we were focusing mostly on this structure, which is the octamer of histone H2, H2B, H3, H4, surrounded by 147 base pair, and studying how modification of this structure could impact the repair pathway mechanism. DNA double-strand breaks are actually the most toxic lesions, and we are mostly focusing on studying how those lesions could be repaired, because when those DNA lesions are not properly repaired or unrepaired, they could lead to apoptosis, and so increased cell death, or they could also lead to mutation, deletion, insertion, which leads to genomic instability, which is a hallmark of cancer. Among sources that creates DNA double-strand break in our cells, we are actually focusing on one of them that is endogenous, which are the replication stress. And when we call replication stress, is everything that can block the replication fork to progress during synthesis of the DNA. And as long as we keep those block of the replication fork, it could lead to DNA damage and DNA double-strand break. Fortunately for us, those DNA double-strand breaks are going to be repaired, but everything is happening in the context of the chromatin. As you can see here, the chromatin that surrounds the breaks is going to be modified to be able to repair those kind of breaks, starting with the phosphorylation of the histone variant H2AX, leading to the well-known mark gamma H2AX. And this mark is going to lead to the recruitment of different enzymes. And the one that interests us today is the RNF168, which is a E3 ubiquitin ligase that is responsible for the monoubiquitination of the histone H2A on its lysine 13 or 15. And why is it important to understand this modification is because this is this mark of epigenetic that leads to the recruitment of different repair factors that are involved in different repair pathways. And as the choice of this repair pathway is regulated within the cell cycle, it's important to make the good choice of repair to avoid genomic instability. And then as all those repair factors are recruited to the break by recognizing the same mark on the histone H2A, we were wondering how the recruitment of these factors is regulated. And that's why we came with the hypothesis that the histone variants that could replace the canonical histones could be the regulator of the RNF168 dependent signaling. And we actually focused on the histone H2A family, which is the most heterogeneous family of histones, containing lots of isoforms such as the H2AX, the H2AZ, the macro H2A, which are specific histones as they contain non-histone domain named macro domain, and small H2A that were interested for us because they don't contain lysines that should normally be ubiquitinated by RNF168. And so for the purpose of my project, I first studied the histone fold domain and avoiding at first the non-histone domain to be able to see how those histone domain could impact the signaling by RNF168. And so to answer those questions, I actually used a reconstitution of nucleosome core particle where I've purified recombinant histones, the canonical one, H2, H2B, H3, and H4, but also the H2A variant that were all his-tagged to be able to recognize them by using the same antibody. I then refolded octamers, and using a specific DNA that can bind to this octamer, I was able to reconstitute either mononucleosomes or dinucleosomes as shown in this native gel, where we can see that the delay of the DNA on gels is actually due to the presence of the protein that are stained here with the instant blue staining. And using these nucleosomes, I also purified enzymes that are responsible for the ubiquitination of those nucleosomes, and our favorite E3 ubiquitin ligase, RNF168. And as you can see here on this Western blot, when I do this reaction on the canonical H2A, I have a nice ubiquitination of this H2A. I did the same with the histone variants, and as you can see, as the H2A bar body deficient doesn't contain lysine, it cannot be ubiquitinated. But then if we focused on the H2AZ and macro H2A depleted in macro domain in this reaction, we can see that those variants are more efficiently or more rapidly diubiquitinated by RNF168. And this is probably due to the fact that those variants have a longer acidic patch, as shown here with the H2AZ, or a more exposed acidic patch to the surface for macro H2A. And actually, we validated this result by just mutating these residues on H2A to have the same residue than H2AZ, and we were able to recapitulate this more rapid diubiquitination of the nucleosome, meaning that the acidic patch in this variant seems to be more more helpful to guide the ubiquitination reaction by RNF168. Then I was interested in understanding how the repair factors could be recruited to this mark. And so I've purified all the minimal domain of interaction of this protein that can recognize the ubiquitination of H2A. And I used them in pull down assays where I bound them thanks to the specific tag. I then came with mono-ubiquitinated nucleosome. So as you can see here, all the variants were mono-ubiquitinated to be able to assess the impact of the variant itself. And I then put them in pull-down assays. And as you can see here, where we have ubiquitination of H2A, we have a nice pull-down of all those repair factors that are actually interacting with the nucleosome. I did the same with the bar body deficient that cannot be ubiquitinated or without the ubiquitination reaction. And as you can see, we don't have any binding of the repair factors. When we do the same experiment with the H2AX and H2AZ, we don't see any differences in comparing with the canonical H2A. But then when we look at the macro H2A depleted in macro domain, we don't have binding of, we don't have any interaction with the 53BP1 and RAD18 factors. And this is not due to the fact that I depleted the macro domain, because once I reconstituted the full-length macro H2A, I obtained the same result, meaning no interaction with RAD18 or 53P1, meaning that this variant, macro H2A, seems to be the only one of the H2A family to discriminate for the binding of 53P1 and RAD18. I then looked further in vitro using a cell model where the macro H2A 1.2 specifically is knocked out in U2S cell lines. And I actually express all those minimal domain of interaction with the ubiquitinated nucleosomes of the repair factors. And so here we have RAD18 and 53P1. And as you can see here, when we specifically treat cells with a replication stress inducer, the HU treatment, we can see that in the absence of macro H2A, we have an increased foci formation of the RAD18 and 53P1 that are now actually able to bind the nucleosome as we don't have macro H2A. And if we look at factors that can bind macro H2A, when we don't have macro H2A in these settings, we don't have any differences in the foci formation of those factors. We then took advantage of our model where we can actually abolish the ubiquitination of these variants, macro H2A, by actually mutating the lysine 11 on macro H2A. And as you can see on this Western blot, just by mutating these lysines, we actually lose the ubiquitination of macro H2A. And using this model, we actually we tried to rescue the experiment shown previously. And as you can see with the wild type version, we are able to rescue the RAD18 and 53P1 foci formation. But with the K11R mutant, we are not able to rescue this increased foci formation, meaning that it's the ubiquitination on the histone macro H2A 1.2 by RNF168 that is important to restrict the binding of RAD18 and 53P1. But then we looked also with the endogenous protein, and we actually had the same results with the HU treatment, so replication stress. without macro H2A 1.2 in the cells, we have an increased foci formation of rad-18, and this is not rescued with the K11R macro H2A. Then we decided to use another model to induce replication stress that was actually published in Philippe Oberdorfer teams in 2018, using the U2S265 cells that actually express a LACO array, and we can come with a construct M-cherry LACAR to create a block of the replication fork that can be visualized by foci just showed here. And when we look specifically at cells that are under replication and where we put the M-cherry LACAR block, we can see that we have an increased incorporation of macro H2A 1.2 at those LACAR foci as shown, as it was shown in this paper. Interestingly, when we look at the recruitment of repair factors in this model, we can see that we don't have a recruitment of rad-18 at those LACAR where we have macro H2A 1.2. But once we deplete this macro H2A 1.2, we can see that we have a nice recruitment of rad-18 at those LACAR. And this is actually followed by a reduced recruitment of BRCA1 and an increased gamma H2X signal. If we try to rescue this experiment using our wild-type or K11R mutant of macro H2A 1.2, we can see that we can nicely rescue the recruitment of BRCA1 either with the wild-type or the K11R mutant, meaning that BRCA1 recruitment at the replication fork is independent on the ubiquitination of macro H2A 1.2. And when we do the same for rad-18, we cannot rescue the accumulation of rad-18 at the LACAR when we don't have ubiquitination of macro H2A 1.2. And what's very interesting is that when we are not able to rescue this accumulation of rad-18, we can see that we still have a high proportion of gamma H2X signal. And so the next question we had is it because we have an accumulation of rad-18 that we have this high amount of gamma H2X signal? And so we decided to co-deplete both protein rad-18 and macro H2A. And as you can see in here, when we co-deplete macro H2A 1.2 and rad-18, we actually rescued the gamma H2X signal at the LACAR, meaning that the accumulation of rad-18 could lead to more gamma H2X signal in this context. We went further with the survival of the cells and the lethality of the H2 treatment. And as you can see with this clonogenic assay, when we don't have macro H2A, the cells in blue here are more sensitive to the H2 as much as without rad-18 in U2S wild-type cell lines. But then if we do the SI rad-18 in the knockout macro H2A 1.2 cell lines, we can rescue the survival of those cells, meaning that the presence of rad-18 in the knockout macro H2A 1.2 and the accumulation of rad-18 seems to be toxic in cells that are under replication stress. And it seems to be specific to replication stress, as I did the same result with another replication stress inducers, the acetylcholine treatment. And you can see that we have the same phenotype, meaning that when we deplete rad-18 in the knockout macro H2A 1.2 cell lines, we rescue the survival of those cells. And I did the same experiment with another treatment which caused DNA double-strand breaks, the irradiation, and we don't see any phenotype using this way of creating breaks. Moreover, I also checked if it was dependent on the ubiquitination of macro H2A 1.2, and as you can see, we can only rescue the phenotype of sensitivity using the wild-type and not the K11R mutant, meaning that the ubiquitination of macro H2A 1.2 prevents the rad-18-dependent toxicity upon replication stress. So far, what we showed is that RNF168 is capable of ubiquitinating a macro H2A 1.2, and this ubiquitination acts as a barrier for the recruitment of 53p1 and rad-18. Moreover, this ubiquitination seems to not be important for the recruitment of BRCA1 at those loci. Then the next question we had is that as macro H2A 1.2 is mostly found in the heterochromatin and it caused the compaction of the DNA, we were wondering if this function could be also important in this mechanism to respond to the replication stress. But so far, what we showed is that in the absence of the ubiquitination of macro H2A 1.2, we have an accumulation of rad-18 that leads to more gamma H2X signal, and so potential genomic instability, and more toxicity as the cells are more sensitive to H2 treatment. And so what could be the leading cause of this toxicity? Actually, radiatin is an important protein that is involved in different repair pathway. And as it was shown for other repair pathway, when we choose the wrong pathway at the wrong loci, it could lead to genomic instability. And as radiatin is involved in all those DNA damage tolerance pathway, we actually focusing on trying to understand which pathway could be the cause for the toxicity led by radiatin accumulation. And so far, our results seems to be to point towards fork reversal to be toxic in those loci. And with that, I'd like to thank all the members of my lab, specifically Andrian and Elise, as well as Elodie. I'd like to thank all the collaborators, the fundings, and of course, I'd like to thank you for your attention.
Thank you, Maxime. I just want to remind people to put their questions in the chat, or sorry, not in the chat, in the Q&A. And we actually already have a question in there. So it says, sorry if you mentioned this already, but is the diubiquitination of H2AZ due to double ubiquitination at the amino terminus? Or perhaps is it combination of amino and C-terminus ubiquitination? That being said, how are you controlling for C-terminal ubiquitination of H2AZ or H2A, which is very prevalent?
That's a very good question. So so far, what we know is that on H2AZ, there should be only one amino acid that is ubiquitinated by RNF168, which is the K15 in H2AZ. But I've not done any mutant in the C terminus to see what could be the lysine that is diubiquitinated. For now, what I can say is that it's certainly as the enzyme I'm using in the in vitro reaction are only doing monoubiquitination. It's certainly two monoubiquitination that I have in this H2AZ variant. And then can you repeat the second part of the question, please? Sorry.
I mean, it said, how are you controlling for C-terminal ubiquitination of H2AZ or H2A, which is very prevalent?
Yeah, actually, I did not look at the C terminus of H2A, but what I can say, for example, is that I did a chimeric construct in H2A where I just replaced so in H2A I have only monoubiquitination, and when I replace the 16th amino acid of the H2A by those of macro H2A, so the 16th amino acid in the N terminus, I actually have this diubiquitination happening. So it may be only in the N terminus.
Okay, thank you. So we have another question. So is ATR involved in the recruitment and function of RNF168 at replication stressed forks and the repair?
That's a very good question. And actually, unfortunately, we haven't looked at ATR yet, but we are certainly going to do it later.
Okay. And another question is, what about the dynamics of the time course of the reaction? Is it consistent with the in vivo course of events?
I'm not sure to understand the question, but I'm gonna try to answer it. So we've not looked at the dynamic of the ubiquitination in cells. What I know is that I actually did an experiment showing that when we do the H2 treatment, macro H2 1.2 is more ubiquitinated on its lysine by RNF168, because as I deplete RNF168 in this setup, I'm losing the ubiquitination of macro H2 1.2. But we've not looked at the dynamic of the ubiquitination in cells right now.
Okay. And just one last question that we'll do live, and then if any others come in later, you could answer them by typing into the Q&A. But so the last question is, did you see or do you think there is a difference between the recruitment of macro H2A 1.2 at replication stress sites in euchromatin versus heterochromatin?
So that's a very good question. And actually, our results seems to be like really specific to certain loci. So what we're thinking right now is that it is mostly happening at common fragile sites where the replication is more difficult to be processed. And so we are actually mostly focusing our future research in understanding which loci could be targeted by this macro H2A 1.2 incorporation.
All right. Thank you very much, Maxime, for sharing your work today and for an engaging talk. So we'll move on to our next speaker. Our next speaker is Jean-Philippe Lambert, who is an associate professor in the Department of Molecular Medicine and Cancer Research Center at the Université de Laval in Quebec, Canada. Dr. Lambert is an expert in the use of functional proteomics and mass spectrometry-based techniques. His research program aims to better understand the loss of transcriptional flexibility found in numerous cancer cells and how this change can be targeted for clinical benefit. And the long-term goal of his research program is to obtain a detailed mechanistic understanding of the transcriptional regulation of individual genes and how this is impacted by cellular contexts. So Jean-Philippe, please take it away, and we're excited to hear about your work today.
Thank you so much, Jennifer, for introduction and for the presentation and for the opportunity to present my work today. Can everybody see my slides properly? Yes.
It looks good. Yeah.
Awesome. So in my group, we've been really interested in understanding how various signaling pathways really converge on a number of writer, reader, and eraser modules at the level of chromatin to regulate both transcriptional activity, but also more generally chromatin itself. So this is like, as for this crowd, everybody knows how, and Maxime introduced that very expertly as well, how the nucleosome, really the fundamental subunit on chromatin, is really heavily modified by a number of these molecules, including that leading to the acetylation, methylation, bucosylation, and phosphorylations of a number of residues. And in recent years, really, there's been like a really large array of effort that have been targeted and to really target these either writer, reader, and eraser modules for clinical benefit. So I'm showcasing here a number of these efforts targeting either the acetyltransferases themselves, especially CRAB-BP300, histone deacetylases, metal transferases, or even chromatin remodeler themselves, such as at the SWI/SNF subunits. So in the lab, we've been particularly interested in acetylation of lysine residues for years now, and with a particular focus on the reader of these marks, which are done principally by a bromodomain protein. So our interest in really focusing on understanding better the biology of bromodomain containing proteins is derived from their essentiality and in a number of cancer subtypes. So if we look here at the data, then this is from the deep map data, where they systematically screened for essentiality of in over 1,000 cell line now by CRISPR-Cas resistant screen. We can see that there's quite a few of the bromodomain containing proteins, including BRD4 and others, are essential in all, if not most, cancer cell lines, and those that are shown in blue here, and there's 15 of them, that are really selective for specific cancer types. So by targeting bromodomain containing protein, we can actually effectively prevent the growth of quite a few cancer type in vitro, at least. So bromodomains, like as I mentioned, are the main reader of acetyl lysine residues. Here I'm showing you a crystal structure of the first bromodomain of BRD4. We can see it's composed of 4 alpha helix with the link with 3 loops, and this is a side view on the left and the top view on the right. And what we can see is that it forms this deep hydrophobic pocket right here in the middle that I'm highlighting, where the acylated substrate here, it's a histone peptide derived from histone H4, acylated lysine 12 and 16, where it fits. One mark can fit really nicely within that pocket, and other part of the peptide can contribute to the binding in here, in this case particular, lysine 16 on the surface of the bromodomain. So for over a decade now, there's been really strong efforts in understanding how bromodomain recognize their target, but also at using them as a therapeutic target. And this has been exemplified by the molecule on the left here, which is called JQ1, that's been described in 2010 by a collaborator of mine. where this molecules fits really nicely within that hydrophobic pocket here. Again, this is the bromodomain of BRD4. The first one, we can see that the JQ1 molecule really fits within that hydrophobic pocket, preventing the binding of the peptide here, again, of histone H4 diacylated lysine 12 and 16. So these molecules are really effective at disrupting protein-protein interactions, both in vitro and in vivo. So over time, there's been really large-scale efforts, both by academic labs and especially from the Structural Genomic Consortium in Toronto and in Oxford, to develop these probes, but also by private pharma. And now, and this is already a few years old, we have good quality chemical probes and bromodomain inhibitors targeting more than half of the bromodomain encoded in the human genome. And many of them are effective in vivo, both in animal models, but also they've transitioned now to quite a few clinical trials. So we have quite a few tools and compounds that we can explore the biology of these molecules and hope to potentiate the cancer treatment in many instances. So if we focus on bromodomain-containing protein, and especially here, I'm showing you on the very top, and the schematic of the architecture of BRD4, which is kind of the most well-studied protein in the human genome of the containing a bromodomain. So if you focus on BRD4, there's two bromodomain near its end terminus, shown here in green. And then we have other domains as well. So bromodomain, like I mentioned, are domains that are known to bind to acetyllysine residues, that's just shown right here. In the case of BRD4, but also of its similar protein from the BET family, BRD2, 3, and BRDT, these bromodomains are linked by a flexible region, this hinge. And we've shown previously that this can adopt a wide array of orientation, allowing some flexibility between the two bromodomain here. Near the middle of the protein, we have this ET domain, which is a bundle alpha helix that allows for protein-protein interaction to occur here. This is an NMR structure of BRD4 ET domain with a viral protein MLV, which hijacked BRD4 during its infection cycle, shown here in orange. Then we have really extensive regions that can be modified by a wide array of, in this case, kinase. So I'm showing you here in this section over 20 CK2 motif, phospho motif that are intercalated, which are, we believe, impacting BRD4 function. And in the case, again, of BRD4, we have the C-terminal motif that allows its interaction with the PTFB kinase complexes. So we have this extremely complex protein here with numerous domains that allows it to mediate protein-protein interactions. So over the years, one of the questions we've asked ourselves is how do we effectively map the interactome of these protein? And with our overall working goal here is defining how, if the targeting of their bromodomain will impact these interactomes. So to do so, we've been working on optimizing various biochemical protocols to make them functional at the level of chromatin. So this is work that began quite a few years ago now, and during my own PhD at the University of Ottawa in the lab of Daniel Figuez. And what we've developed was a biochemical mean to really gently lyse cells and treat them with both sonication and benzonase treatment to really release in a very gentle fashion under low salt condition chromatin and the associated complexes. And so when we did that, we were able to maintain the protein-protein interactions, but also the protein-DNA interactions under native conditions, and then we can purify them and identify the interactome using mass spectrometry in very effective fashion. So one of the downside of this protocol is that it led to much more background and much more non-specific interaction also being present, DNA being extremely charged, as we all know. So what we've done is we've coupled this biochemical protocol with a bioinformatic one where we can take a protein of interest digest into peptide using a protease of choice, usually trypsin, and then perform LC-MS analysis and score or purification against negative control. And the goal here, and this is a tool we use, that I've been using for many years, called SynExpress, to do so, that allows us to identify proteins that are overrepresented in our purifications compared to control ones. And ultimately, we can visualize these in various ways. Here is a dot plot, which we'll see many times during my presentation, but it can also be done using more traditional means, such as a heat map, for instance. So we've applied this to the bed protein family, composed of BRD2, 3, 4, and BRDT, BRDT being a testis specific, but we tested it here for completeness. And then we perform a time course treatment with JQ1, which is this pan-bed bromodomain inhibitor that targets both the first and second bromodomain of BRD4, as well as BRD2, 3, and BRDT. And then we use here a very quantitative DIA approach for mass spectrometry, which I'd be happy to discuss if needed at the end. But suffice to say that it allows us to have a fully quantitative approach here to quantify our interactome. And what is shown here in this heat map is that the resulting spectral count, as when we apply the MSDIA approach, and here the darker color represent the higher abundance. And then we could observe really readily here in the heat map 3 general behaviors. If we look near the left of the screen, we can see that some protein-protein interaction were reduced when we added our bromodomain inhibitor. So we can see strong colors going to much weaker. Some interactions were mostly unchanged, as you can see in the middle right here. And some were actually, the complete surprising to us, were enhanced when we treated ourselves with the JQ1 inhibitor, as showcased here, for instance, and in other places as well. So if we summarize this data in a single slide, so this was a few years of my life. So what we've learned from this data is that we could quantify about 367 protein-protein interactions that were decreased in this work. And using this information, we did quite a few things. But for instance, what we did is we identified a new substrate binding mode for the first bromodomain of BRD4. So here is a crystal structure generated by my colleague Panagis, where in blue is a peptide that's diacylated from histone H4 at lysine 5 and 8. And then in brown or golden here, we can see that it's a peptide where there's the binding is enhanced by the presence of a tyrosine at the +1 position. So there's an acylation at lysine 221 of BAD1B, and the presence of this tyrosine at the +1 position strengthened the binding to the bromodomain. So for the protein-protein interaction that were unchanged, and there were about 250 of them in our data, using that we were able to identify small linear interaction motif or SLIMs for 2 types of substrates, so the BRD9-like, and as well an NSD2 and NSD3-like substrates. So these are short motifs that interact with the ET domain present on bed proteins. And lastly, for the interactions that were enhanced in our data, and there was about 250 of them again, we were able to use this data to understand better the role of BRD3 at the level of the ribosomal DNA clusters in the nucleolus. And we found a repressive role for BRD3 at these sites in general. So this was all published. And this, so we really, from that information, we learned a really large amount of new data. But so one of the questions we had derived from all this work is whether we were able to gain like a more global view of the local chromatin in itself that's associated with the BRD-containing protein. And for this, we turned to a different biochemical approach, which is the proximity-dependent bioaccumulation assays, or more simply BioID, that it's been known and over the last few years. So in BioID, what happens is that a protein of interest, or a BAIT, is fused to an abortive biotin ligase. It could be, traditionally it's been BRA that's been mutated, and that was done in 2012 by the Roux et al. But ever since, there's been a flurry of activity to engineer these biotin ligase and make them more potent, more rapid, more specific. But what they all had in common is that they allow the catalysis of the activation of biotin into its active intermediate form, but they lack the capacity to retain it and to direct it to a particular site. So usually biotinylation in cells is a very targeted approach, and in the case of BioID and in all of its derivative, that second part, the targeting, is lost. So what happened is that the activated biotin starts to slowly diffuse away from the bait of interest, and it forms this cloud of activated biotin, which can then react with exposed lysine residues, the amine on the epsilon amine on the side chain of lysine, and covalently mark them with biotin. So this can be done in very quick fashion or in a very slow fashion, depending on the goal of the experiment. And then at the end, we can purify these proteins using streptavidin beads, and then use a traditional mass spectrometry-based approach that I've described before quite effectively to quantify these proteins and then filter them against controls and other things, and to obtain a good level of understanding of the proximity interactome over beta of interest. So this has been, since the early effort we've led in that field in 2015, where we were targeting histones and others, there's been quite a flurry of work of using BioID and its derivative at the level of within the nucleus itself, but also at the level of chromatin. And then here, this is from a 2020 review. As it shows, it's been shown to be quite effective at the nuclear pore, the nuclear membrane, in the nucleus. We've done that ourselves, and the actin, telomeres, centromeres, nuclear body, speckles, and others. But at the level of chromatin itself, we've done histones, transcription factor, DNA repair factors, chromatin remodelers, and quite a few. So this is a very flexible approach, and that yields extremely good results in most cases. So it's quite worth thinking about it if you're interested in specific factors. So in our hands, so then we were equipped with these two techniques. So we've deployed them for the whole family of bromodomain containing protein, and I'm showing you here the 42 human protein encoded in the genome with at least one bromodomain, and that they've been classified based on this scheme we established a number of years ago based on the structural homology of the bromodomain themselves. And that what we've shown here in this bar graph in red is the amount of protein-protein interaction detected by using a traditional affinity purification-based biochemical approach, and in blue, this BioID-like approach. And what was really interesting, for one, is that it's very, despite using the exact same constructs, this yielded very different results in terms of amounts of protein-protein interaction detected. In general, BioID was a bit more effective, but it was still, both approaches were actually quite successful in our hands. And then if we generate one of those hairball or this network of interaction with protein-protein interaction, what we can see is that there's really a divergence between the type of protein interactions that can be quantified using both approach. These are very both approach yield data that can be integrated to have a larger depth of information. And that was work that was spearheaded by Lucien in the lab, a PhD student in my lab here in Quebec City. So just looking at to gain a better understanding of the modular organization of within this data, what we've done is we've applied a clustering where we look at the profile of protein associated with a given bait protein in our BioID data, and then the resulting cluster allow us to really refine our understanding to a fair amount. So here, for instance, I'm showing you 8 different clusters, and then these are what their compositions are, that are generated using this automatic clustering approach to better understand how the various proteins are organized at the level of chromatin. So from this, what we were really interested in is to define really what is bromodomain dependent within these clusters. And for this, we decided to focus on one specific family, the mammalian SWI/SNF complexes. So I'm showing you here just an overview of the 3 types of mammalian SWI/SNF complexes present in the human genome. these have been extensively studied, and in our case, our interest was derived from the fact that these are the complexes that contain the larger amount of protein with bromodomain themselves, and also the greater amount. So there's 5 different subunits shown here in dark, in bolded letters, and in total, there's 10 bromodomain that are present within one of or all of these complexes. So there's kind of 3 flavors. There's the canonical BAF complex, here shown on the left, the polybromo BAF in the middle, or PBAF, and then on the right, the non-canonical BAF complexes. So you can see that they all act like clamps around the nucleosome, and with one bromodomain within the ATPase subunit, either SMARCA A2 or A4, so BRT1, BRM, and then the other bromodomain being present in either BRD7, polybromo, PBRM1 that has 6, or BRD9 as well. So what we did in the lab, and that was efforts that were spearheaded by a recently graduated PhD student, Linda Agbo, in the lab, is we wanted to better understand the organization of this complex. So what Linda did is that she first established an assay where she used one shared subunit, SMARCA CD1, which is shown here in blue, and then performed BioID from it. And then doing so, we're able to capture protein interaction from most of the SWI/SNF, mammalian SWI/SNF subunits in a single experiment. So once this was verified to be functional, what she did is that we engineered CRISPR-Cas9 knockout cell lines where we knocked out every single subunit that had bromodomain within SWI/SNF complexes. So BRD7, polybromo, BRD9, SMARCA A2, SMARCA A4. And then we perform again our BioID here using SMARCA CD1. So these are dot plots that we call these plots right here. So here the color of the circles corresponds to the absolute, it's an absolute quantification measure. So in this case, this is a spectral count. So the darker it is, the more abundant the protein is. The size corresponds to the relative abundance for a given protein. So in this case, for example, SMARCA CD1 across all of our experiments. And the outer edge, and it might be a bit easier right here, the color of the outer edge corresponds to our false discovery rate. So this is how much of an enrichment is there for that particular protein compared to our controls. And those that are marked with asterisks are just what we use to purify here, in this case, what was tagged with the BioID fusion. So doing that, what Linda observed is that, unsurprisingly, when we knock out BRD7, we lose BRD7, but we also lost PBRM1. And that was known at the time based on the overall organization of the complexes. What was much more surprising to us was this concurrent increase in non-canonical BAF subunits, namely BRD9 right here. So when we lost one part of the complex, we tend to favor another one of the subcomplex of mammalian SWI/SNFs, and it's non-canonical BAF in this case. So what Linda did was to further this experiment, then she just moved or tagged around to either polybromo, SMARCA A4, or BRD9 in our parental or BRD7 knockout cell lines. And again, if I could just bring your focus right here at the bottom, here again, we've said the reverse, where BRD9 was strongly associated with the bed protein, BRD234, through its small linear interaction motif we had previously identified. And then when we knock out BRD7, this was very much reduced because at this time, BRD9 was more associated with the mammalian SWI/SNF complex itself. So based from this data, we were really interested in understanding is the modulation of the mammalian SWI/SNF interaction really altered by bromodomain inhibitors? So we quantified that. So again, we turned to BioIDs. In this case, we tag either SMARCA A2, A4, or polybromo, and then we treated them with either DMSO-controlled PI3 bromodomain inhibitor, which targets the bromodomain of SMARCA A2, A4, and the fifth bromodomain of polybromo, or JQ1, which doesn't target these complexes directly, but rather the BEP protein, as I mentioned. What was really interesting here is that JQ1 was the one that had the most pronounced effect. So that was very much surprising. So we really wanted to further more. So what Linda did is she started to investigate BRD7, this idea in a specific breast cancer subtype. So in invasive lobular breast cancer, so these are tumors that develop in the lobule of the breast. What happened is that this is tumor type that are defined by the loss of CDH1, which is shown right here from the TCGA data. So in ILC, CDH1, which is encadherin, is lost in the vast majority of these tumors. And what Linda observed is then digging through the genomic data is that the BRD7 levels are much more, are significantly reduced as well in these tumor type, and this is a significant co-occurrence. Look, digging into this a bit more, what we have observed is that these kind of tumors are usually occur because of the loss of the 16q chromosome arm, where CDH1 is lost concurrently with BRD7 because they're relatively close on this chromosome arm. So then what Linda did was able to show that indeed, when cells are lacking either BRD7 or polybromo, these are cells that are sensitized to pan-BET bromide inhibitors such as JQ1. We tested a few, but we're just showing JQ1 right here. So there's a potential here to repurpose the BET bromide inhibitor in these BRD7 deficient tumors that we're interested in further. But more generally, we also wanted to know whether BET protein are impacted by bromide inhibitor targeting other readers, as we've shown for mammalian SNF complexes. So, and I'm sorry for this extremely busy slide. So what we've done here is we selected BRD4, and we treated these cells with either DMSO control, JQ1, as we've done before, or other type of bromide inhibitors. So newer generation of BET bromide inhibitors. So 075 here is just much more potent than JQ1. 744 here is selective for the second bromodomain of BRD4. The CBP30 and targets PrEP-BP and EP300. The BI9564 targets BRD9, and PA53 I've already introduced targets mammalian SNF complex. And what we've shown is that these, so if we look just at histones at the bottom right here, most of these compounds will reduce BRD4 interaction with histones. There seems to be some residual impact of these compounds toward BRD4 with regard to their more cognate substrates, so histones themselves. So if we look for a number of partners that are on chromatin itself, we can see that these are mostly affected by the molecules that target BRD4 directly, shown here in the light blue. There are a few bizarre, weird events that are occurring. So these are, if we look right here, JQ1, and that's been reported in one bioRxiv paper last year as well. So it seems to form neo-interactions with both members of a centromere and as well the cytoskeleton. So these are novel interaction where the compound allows for a new binding surface to form and potentially leads to these interactions. And then if we look for our mammalian SNF subunit, again, we see that we seem to impact polybromo more deeply with compound targeting the BET protein themselves. And just a quick note on this one, there is residual activity toward BRD4 right here. So this is why it's also reduced. So what we've shown here is that the BRD4 and BET proteins themselves really seems to be at the central node of chromatin in terms of bromodomain inhibition. So targeting them seems to have the most pronounced impact in all of our assays. So to further that a bit more, we turn to our favorite model in the lab, which is melanoma. So melanoma is a cancer type that's still poorly managed in Canada, and that's whose incidence is still ongoing. So I'm showing you here a patient that has a very extremely metastatic disease, and initially he was treated with this a number of years ago when these new inhibitor targeting BRAF, mutant BRAF, which is the most common alteration in these patients, and you can see how he responded really, really effectively for the first few months. But then unfortunately, as it most often happens in melanoma that are metastatic, resistance occur really quite rapidly and tumors grow back at the exact same location. So these compounds are only good for a few months of treatment. So more recently, checkpoint inhibitor, so targeting either PD-1, PD-L1, and others, have started to emerge as well in the clinic, and these have for the last 10 years or so, and these have yielded better results. So you can see here after 4 weeks, the tumor is shrinking, and then over time it goes back, it goes away, but there's still some tumor growth here, even after a full year. So the patient is still alive, but there's still unmet clinical need here where these patients are not cured. And realistically, for the most advanced melanoma cancers, the overall survival of over 5 years remains quite low, around 25–30%. So our interest in melanoma was derived from the fact that, and that's been published for about a decade now, that both BRD2 and BRD4, and I'm showing you the raw data from these publications, that the levels of BRD2 and BRD4 correlates really nicely with disease progression in melanoma. So from melanocytes, which are the pigment-producing cells in the skin, to all the way to metastatic tumor, you can see how these levels are increased in both cases. So we use melanoma as our model to further test bedroom domain inhibitors. And what we've shown is that realistically, the only in these cells, the only compound that are effective at decreasing MYC level were the one targeting BRD4 directly, and that was occurring concurrently with an enhanced level of activation of the p53 pathway, leading to accumulation of p21. At the same time, what we observe is that the phenotype of these cells was drastically altered. So these cells change from a very small, mostly rounded, proliferative type phenotype to an elongated, differentiated, more melanocytic cell type. And then when we tested all of our compound, what we observe again is only the one targeting. This change was only occurring for the one targeting BRD4 directly. So again, the BET proteins and BRD4 in particular seem to be really the central node of transcriptional regulation in these cells. So one of the questions we had is, can we better understand chromatin remodeling more generally in these cells? And for that, Lucien developed a new tool that we call the Cas-Ultra. And this is derived from pioneer work done in the Carr lab and others, where we fuse, and this is shown in the middle here, an inactivated Cas9 to an ultra-ID fusion, the moiety. And then we use that to target it to particular loci on DNA here, the MYC promoter. And then our idea was to do so in the presence or absence of treatment to see how the chromatin itself was remodeled. So Lucien benchmarked this initially at repetitive regions, and it worked really well for centromeres and telomeres, and I'm showing you data here. This is also available in bioRxiv. And then when we targeted the MYC promoter itself using this approach, we've seen, and this is a bit difficult to read on the screen, I'm sorry about that, quite a few of the mammalian SWI/SNF complexes were reduced as well in following treatment with JQ1. So these are they're enriched with our control treatment. So again, we seem to be, the bedroom domain inhibitor seems to be really remodeling profoundly the composition itself of chromatin at specific loci, going beyond just removing dead proteins from their histone targets. So we're still pursuing this data further. So this is very much an ongoing studies and efforts on our part. So as I mentioned, so bedroom domain inhibitor are now in the clinic. There's over 50 clinical trials report involving them. So far, the data that's been published from these trials have revealed that the inhibitors are generally well tolerated. They do lead to disease control and partial response in a number of tumors, but usually this comes with a narrow therapeutic window. So our goal now is to kind of potentiate how we can use these compounds by combining them with metabolic alterations. And I'll quickly go over this for the remainder of my presentation for the next few minutes. So as you all know, and this is not going to be a surprise to anyone, so the acetyl-CoA is the necessary cofactor for acetyltransferases that leads to acetylation of lysine at the epsilon residue that's shown right here. So for those that are not metabolically inclined or forgotten their initial biochemical schooling way back when, so acetyl-CoA is mostly derived from glucose in mammalian cells. And the way it works is that usually, so glucose is imported in the cell, and then in the mitochondria, acetyl-CoA is generated, but this cannot be exported away from the mitochondria directly. Rather, it needs to be converted through citrate and then exported as citrate into the cytoplasm and nucleus, where the enzyme ATP citrate synthase actually can convert it back to acetyl-CoA, where it can be used either for lipogenesis or reactions such as acetylation of lysine residues, which are shown here. So what Naomi and PhD student in the lab and Jeremy research associate in the lab have done is they've established a model where we were able to knock out actually in cells, which is usually non-viable. But here what we did is we generated, we allow cells to survive through the supplementation with acetate, which engaged a different pathway involving acetyl-CoA synthetase, ACSS2, which allows the conversion of acetate to acetyl-CoA, and then again, lipogenesis, acetylation, and other reactions. So if we look in cells, what we do is if we grow these cells with media containing dialyzed serum, so there's a small amount of acetate in normal serum. That's why we use dialyzed serum here. So these cells grow extremely poorly and are not very viable in the absence of supplementation. When we treat them with acetate, these cells grow extremely well. So we have a very easy model to manipulate and work from. And then what happened is this: if we remove in these knockout cells, if we remove the supplementation for us in a time as short as 10-15 minutes, we can really abrogate acetylation very profoundly in these cells, which is shown right here. So to get an idea of how lack of acetylation was remodeling chromatin in these cells, we turn to our trick with turbo ID tag histone protein. And again, the idea was to use those cells in the presence or absence of acetate. And what we've observed is that there was a general depletion of many components, including those that had bromodomain from chromatin here on the left. So this is comparing in the same cell line turbo ID without supplementation or with here on the left. So viewing that as a dot plot, we can see there's a general reduction between the presence of absence of acetate. So to really further investigate this behavior, we selected BRD9, which was poorly represented in this general assay. So here, BRD9 is, as I mentioned, is interesting because it has both the bromodomain, which we can target very effectively by chemical with inhibitors, and it has a SLIM that allows it to interact with BET directly, and a DUF domain that allows it to be incorporated into SWI/SNF complexes. So when if we purify BRD9 in the presence of acetate, so when there's acetylation, we can see that it forms, this is just a small subset of the interaction that it forms with histone, SWI/SNF, and BET protein. So if we remove the acetate from these cells, an hour later, the association with histones is very much reduced. If we treat cells with bromodomain inhibitor targeting the bromodomain of BRD9, again, the same thing happens, but it also loses interaction with BET protein. And if we target the BET protein themselves, then nothing happens. This is just a very strong association. And then, so here what this thought is that the absence in our trick of just reducing global acetylation was a way to mimic phenocopy to some extent bromodomain inhibitors, as in the case of BRD9. And then when we do simple tests where we look for synergy between the absence of acetylation using, again, our trick of depleting these cells out of acetate in the presence of either a JQ1 or a very non-specific but targeting bromodomain, so bromosporine, which targets most bromodomain in the human genome, we can see that there's a reduction in growth with the synergistic treatment. So, again, what we've learned from this is that there's potential here of using lack of acetylation as a readout, as a way to potentiate bromodomain inhibitors. So we also know that the lack of ACLY in melanoma is actually quite impactful to their fitness. So here again, this is data from the DeepMap database from CRISPR screens. We can see that melanoma cells on the left are particularly sensitive to loss of ACLY than compared to all other cell line tested at that time. So this figure is a bit older now. And then we believe this is in part due to this really central transcription factor in melanoma called MITF, which is an oncogene which allows for both melanocyte biology in normal cells, but also in melanoma itself. So MITF allows for proliferation of melanocells, control the expression of enormous metabolic gene, especially SCD, and is really a key player in lipid synthesis. So what we've shown is that if we look for correlation between the expression level of MITF, the more melanoma cells of MITF, the more they're dependent on the presence of ACLY for their survival. And then what my collaborators have shown is that in melanoma cells expressing high level of MITF, these are extremely sensitive to SCD inhibition, while the cells that don't have much MITF are not. And then supplementation with fatty acid can overcome this limitation. So really, this raises the question that whether MITF allows for if its activity is dependent on acetyl-CoA levels and more generally on acetylation. So again, work with the collaborator Colin Goding in Oxford. What we're able to show is that MITF itself is actually acylated by CREB-BP and EP300, especially there's these two sites within this DNA binding domain right here. So these are two conserved sites throughout the protein family. And if we look especially for lysine 243, it is predicted to be directly engaging the pentose phosphate backbone of DNA here, as we can show right here. So presumably that would disrupt his DNA binding activity. What we actually found was actually somewhat surprising at first, was that yes, indeed, so the modification of MITF at this residue, residue 243, either if we use the mutation mimicking the absence of acetylation, so K2R, or K2, or mimicking the presence of acetylation K2Q, led to a reduced binding affinity. And this was done using single molecular tracking, in this case of MITF. But then when the ChIP-seq experiments were done, what we've seen is that the acetylation of MITF allowed for a general decrease of its binding across the genome, but it allowed it to be concentrated on very specific genes involved in its key function, which were related to, in the case of normal melanocytes, to production of melanin, which is the case for this PMEL gene, or more generally proliferation. And then this is the last slide. So when we turn with collaborators to in a zebrafish model where for fish lacking MITF, when we macroinjected the wild type MITF back in these fish, they could form these pigmented cells quite nicely. So when we macroinjected MITF lacking the capacity, phenocopying the absence of acetylation, there was very few melanocytic cells being produced. And then the one that would mimicking the presence of acetylation, there was quite a bit more. And this is the quantification of this. And then in xenograft model of this, using the same idea, where using 501 male cells that are very poor in graftment, normally in xenograft, we can see that in 10 mouse, there was 0 tumor formation, but when we overexpress MITF, Wild type version, there was some engraftment. The MITF mimicking the acylation on this site was very much 10 out of 10, and then the one mimicking the absence was 0. And if you look at the tumor themselves, you can really see how these tumors are extremely melanocytic, so very much dependent on the MITF itself to form. So we're continuing to work with Colin and others to really investigate how, through a combination of proteomic inhibitors, acylizing modification, we can counteract melanoma and MITF activity for clinical benefit. So just to conclude, and I'm sorry, a bit long. So what we've shown is that the proteomic-containing protein are really key regulators of transcription, and they're emerging therapeutic targets. They've now been studied by quite a few pharma, and there's hope that there's going to be molecule making all the way to clinical use. When we use proteomic inhibitor, we disrupt the order organization of protein complexes on chromatin in ways that are hard to predict. So we disrupt transcription, but it's a much more global event. Metabolic alteration can really significantly impact the epigenome, but also we have some evidence now that it seems to be potentiating proteomic inhibitor in melanoma. And I mean, we believe that functional proteomics is very effective at characterizing chromatin in general, and it's a very good tool to use. So just quickly, the work that I presented today was done in large part by Jérémy in the lab, Naomi, Lucien, and the recently graduated student Linda. With the help, some of the early work was done in Anatole House lab at the Lund Feltenbaum Research Institute. The melanoma work is done in collaboration with Colin Gutting in Oxford. Some of the early crystal and structural biology work was done with Panagis at the Structural Genomic Consortium, and as well as been local support for a number of great labs. So thank you very much for your attention, and I'd be happy to take any question you may have.
Thank you, Jean-Philippe. Please put your questions in the chat. And I guess I have a very general question, just maybe while other people are thinking about their questions. So I remember when JQ1 was maybe in some of the first publications with using it as an inhibitor, it was suggested that all cancer cells were more susceptible to it than sort of normal cell types. Do you think, is that something that's like, do we still believe that to be true? Or like you're showing that there might be just specific subsets of cancer, with specific mutations that are actually much more sensitive to JQ1?
Yes. So yeah, absolutely. So I would say there's definitely, so in general, so there's the most sensitive cells are those that have from this very rare cancer they're called the nut midline carcinoma. So and they're actually a fusion of BRD4, BRD3 with the nut gene. So this is like here that the fusion is a driving event for these cancers. These are excessively sensitive to these inhibitors because they cannot survive without the expression of a fusion protein. So this is kind of the ultimate test, really, to showcase how effective things are. So in that case, this is excessively, these tumor types are extremely sensitive. Beyond that, usually most leukemia, myeloma, and prostate and breast cancers tend to be very sensitive to these molecules. And then melanoma as well in our hands and others, we've shown that they're quite sensitive. A lot of solid tumors are not particularly responsive. So that's what we've been learning for the last year or two from the first batch of clinical trials being published, is that so these were very much open basket clinical trial in many cases where they offered them to quite a wide array of tumor types. And yeah, so not all tumors will benefit from them. That's for sure. The tricky part is that, especially in the case of BRD4, so BRD4 is required for normal, from pretty much most cell type survival. So we need to, that's why we're working on enhancing that therapeutic window to allow greater differentiation between malignant tissue and normal tissue in the treatment, because that's where most of the side effects seems to arise at this point. And especially most stem cell type, tend to be sensitive to these inhibitors. So we need to make the tumor type the tumor cells more sensitive than the patient own stem cells. So one giant progress on that front was development of kind of a third generation of compound. I haven't touched on them just now, but there's now we have really good for the BET protein at least third generation compound, and these are can selectively target the first or the second bromodomain. So if I go back here, so most of the work I've shown today was targeting both bromodomain at the same time. So now work from AbbVie and GSK, they've reported a number of compounds that target either the first or the second bromodomain of these proteins. And what they were able to show from that is that while targeting the first bromodomain leads to a much more potent phenotype, so you have a much more direct response, targeting the second appears to, in preclinical work anyway, hopefully we'll have clinical data sooner than later, seems to be much better tolerated and may allow for a larger window, therapeutic window. So just targeting this second bromodomain. And in the case of BRD2 and BRD4, this is believed because that the first bromodomain, it favors binding to histones themselves, while the second one seems to be favor binding to other substrate as well. So histones, but as well transcription factors and other type of protein. So this might be a bit more effective moving forward on that path.
Thank you. We have a question in the Q&A. So it says fantastic work. I'm wondering, what is the rationale to choose bio-ID, turbo-ID, or ultra-ID in different experiments?
So it really all comes down, I guess that's the perfect slide. So I'll switch that to here. So this is a really good question. So the original bio-ID was kind of was just very easy in a sense that it was slow kinetic. So it took about anywhere from a few hours to over a day to make it work. So the benefit were that, you know, it's the easiest one to do, it's easiest one to control for. Your controls are going to be timed with your samples quite nicely. And it allows you to cover often a complete cell cycle as well. So in the case, so some of the early work we've done with histones, for instance, we were able to get mitosis-specific interactions without having to do arrest and things like that. So it's a bit like, so the slow kinetic can be a benefit at time. And also it allows for a very large accumulation over time of signal. So you, as time passes, you mark more and more of those proteins as shown here on the screen, and this results in just stronger mass spec signal, so just more depth in your experiment. So for quite a few experiments, if you have an unknown gene and you don't really know what it does, it's a very effective way, and I would definitely suggest it. So the other enzymes that are kind of engineered enzymes that have been been designed to be faster, to have extremely fast kinetics going from minutes to an hour or two. Now, usually it comes down to the experimental question. So if you want to do, if you really want to understand like a time course, then yes, like a fast kinetic will be essential. So whether like with regards to the various highly active biotin ligase, we tend to favor now ultra-ID for most of our recent work. And the idea for that is that it's one of the smallest ones. So it's a bit easier to fold, it's easier to express, whether it's in lentivirus or other. And also with regard to chromatin, it lacks a particular domain that has residual DNA binding affinity. So it reduces background. So if you compare ultra-ID to turbo-ID, for instance, turbo-ID will give you like a general background at the level of the nucleolus, which is absent from ultra-ID. So that came online in 2023, for instance. So we tend to favor it now, but from some of our older work, you'll see we'll use different enzymes just based on when they were available. Turbo-ID was available in 2018, for instance. So yeah, so I would tend to favor some of the smaller and faster and less complex domains. So ultralight here doesn't have the capacity to bind to DNA directly, for instance, which is a net positive for us.
Yeah. All right, and another question: how do you correct for non-physiological biotinylations happening during cell lysis?
So during cell lysis, we haven't seen much. So that hasn't been an issue for us because the moment you really crack open cells, and we do so with extremely harsh lysis buffer. So it's we use RIPA with plenty of salt detergent and everything. So there's very little activity left in these cells. So, but the first part of the question with regard to correcting, so what we do is we do the same design that we do for more traditional biochemical purification, but we just do it more to a greater extent. So the idea is to use cells that are expressing the fusion biotin ligase in the same general vicinity that you want to study. So we've used a number of things in the past quite successfully. So we usually do a mixture of like a GFP control where the fusion will disperse throughout the cells. So it gave us like a general view of everything at once to model our background using the same express tool. And then we also compare that, for instance, we usually supplement that with in the biotin ligase used to an NLS element to really force in the nucleus and just give us a general nucleus background. So after that, what we will consider significant is anything that's rising above those levels in a reproducible fashion. So in our hands, that gives us, this is a fairly stringent approach, and we lose some proximity-dependent interactions in doing so, but the resulting data sets are easier to manage because they're much more specific. So we tend to favor, weirdly enough, a smaller list of hits because we find they're much more easy to follow up on than than going for the big number, which then tends to yield quite a few false positive, unfortunately.
All right, thank you. I think we'll stop there because we've run a little bit over time today. So thank you everybody for staying with us. And thank you to both of our speakers today. I'm not sure if there's anything else that any of the other organizers want to say to close out the meeting. If there is, just chime in. Otherwise, I'll just say to everybody, have a great Friday and a really nice weekend. And I think we will see you next time in the new year for the next talk in the series. Sinead, did you want to say anything before people
Yeah,
go?
just to let everyone know that the next talk will be, and the next seminar will be with Sarah Kamens on January 17th. So we hope you all can join us then.
All right. Thank you everybody for joining us today. And thank you to the speakers as well.
Thank you so
Great
much.
talks, both of you. Thank you.